Swayam Prabha. Digital India. Educated India.
Does any one of you have an iPhone? You have a charger? Yeah, because I'm about to run out of battery. You have it or you don't? Okay. No, but I don't have the connector, so no, come on. I mean, it's not a big deal. So you just put it there? Really? How? Miraculously or what? I never heard of that.
For wireless charging then?
No, doesn't matter, but it doesn't matter, don't worry. Go ahead.
Hello everyone, my name is Tejas and today I am going to present on this topic, a naked bacterium emerging properties of a serpome streamlined Pseudomonas putida strain. So the objective in this paper was to edit the genome of the Pseudomonas putida and the specific genome was KT2440. The objective here was this was done because it had properties such as the surface proteins of it were responsible for motion as well as surface adhesion and biofilm formation. So why this bacteria is because it has various properties such as it is non-pathogenic, soil inhabitant and root colonizer. Also it is amenable to a large number of genetic manipulations. Also, the bacteria usually will have surface attachment to specific strains. So it is difficult to attach bacteria to multi-strains. So hence it is difficult, but it can be done with the help of suppressing naturally present adhesins present in the bacteria and then replacing them with the artificial adhesins. So in this paper, they've initially started with identifying genetic determinants. So as you can see in this plasmid, there are total 21 sites present, 21 regions, which are responsible for different kinds of surface properties such as, for example, fimbriae, flagella, or antigen site, exopolysaccharides. So they were focusing on those elements so that they can remove those genes. So totally there were 21 such regions. And the removal was done by homologous recombination. The figure B represents the order in which the regions were removed. And so initially we have this Pseudomonas putida KT2440 and finally the strain is Pseudomonas putida EM371. That 4.7% represents the genome reduction. So as compared to the parent strain, there is a genome reduction of 4.7%. These are the region groups they focused, such as complex structures, surface proteins, regions related to production of extracellular polymeric substances and non-OM associated elements. OM means outer membrane. Some of the properties based on the outer surface membrane such as alginate, which is useful for the bacteria in water limiting conditions. for example, alginate under water limitation stress in order to reduce the water loss in biofilms. Also, it protects against various ROS, that is reactive oxygen species. There was another type, that is curly, which are fibrous elements, curly, that are present on the surface of the bacteria. So alginate and curly and some of the other examples which are present on the surface, they were trying to focus on that so they can make it immobilize. Then in envelope edited Pseudomonas putida, EM371 strain displays a simplified outward facing molecular landscape. As we can see in this figure, they initially extracted lipopolysaccharides which are present on the outer membrane, and they separated them. So they compared parent strain and the modified strain, and this is the SDS page assay. We can see that in the modified strain, which is on the right side, the regular patterns are not present there. So checking differences in the surface exposed proteins, which is also called as SUR form. Small iron oxide nanoparticles covered with carboxymethyl dextran were chemically activated and mixed with intact whole cells. So nanoparticles were used here, and with isoelectric mechanism, they were separated. Parental strain, that is KT2440, 196 proteins were observed, which is shown in the B figure, and the naked strain, which is the modified strain, it had only 12 proteins present on it. Then some of the proteins were present in the parent strain, while some were present, like some of the proteins were present parent, but not present in the modified strain. Also, some of the proteins are present in the modified strain, but not in the parent strain. So how is this possible? So it is because in the paper, and they told that because we are doing the iron oxide nanoparticles, because of the charge distribution is changing, some of the proteins are present in one of the other. Then they checked physiological and morphological properties of both the strains. So both the strains were grown on different carbon sources. As shown in figure A, you can see that the parent strain inhibition zone is larger as compared to the modified strain. So the modified strain is more sensitive. In the B figure, their morphology is shown. It can be seen that both of them have same morphology. C figure shows about the flagella. The parent strain, we can see that clearly that flagella is present there, while in the modified strain, this is the TEM image. A modified strain doesn't have any flagella. And in the D figure, the upper one is of the parent strain, where colonies, we can see that they are pale from white to pink color. Also, they are wrinkled. But in the case of modified strain, it is red color and smooth colonies are formed there. They checked sedimentation, surface hydrophobicity, and membrane permeability properties. So they wanted to check if deletion of flagella is responsible for any sedimentation. So as the parent strain has flagella, it can move and it can swim. So it has properties of moving, but as the modified strain doesn't move, it sediments on the bottom of the flask. So it can be seen here that this shows percentage of cells in solution. Pink color represents the parent strain, while green color represents the modified strain. Percentage of cells in solution means the particles of the parent strain are more in the solution, while those of the modified strain are not in solution, that means they are sedimented at the bottom. They also checked hydrophobicity with math test. Math means microbial adherence to hydrocarbon. So which shows that how much a bacterium is how much bacterium is having affinity towards an organic phase. And lastly, they checked the permeability. So the modified strain, which is the EM371, had more permeable cell envelope. So this CPRG is dye, which is chlorophenol red. It can enter the modified strain, and because of which it is more red as compared to the parent strain. Stress resistance profile of the modified strain. So they checked with different stress conditions, such as they have used some antibiotics and also ADTA, SDS, and hydrogen peroxide. So we can see that in the case of parent strain, the inhibition zone is less, but in the modified strain, it is more. So we can see that parent strain is more it can resist, but the modified strain is more sensitive to all the stressors which are checked here. So it also shows that if modified strain is more permeable, it is more sensitive also. Then auto aggregation of Pseudomonas putida. So they wanted to check if the aggregation is before we checked if the aggregation is happening and the modified strain is going to the bottom, it is getting segregated. They wanted to check if it is happening because of the solids present in the solution. So they checked with different concentrations of NaCl salt from 0 to 400 millimolar, and the ratio of A600 vortex by A600 no vortex. That means the absorbance at 600 nanometer was checked with vertexing and without vertexing. So with vertexing will have flocculation, without vertexing is without flocculation. And the results shown that naked strain, that is the modified strain, showed more aggregation, while parent strain didn't show much. So this is a box Weber plot, and the two strains are represented here. The purple pink one represents the parental, while green represents the modified strain. And this is the ratio of vertex by non-vertex. The more the ratio, that means more the flocculation is there, and green, that is the modified strain, is showing more ratio. That means it is getting flocculated and it is getting segregated. And why is this happening is because why is this happening in case of modified strain is because salting out is happening in that, while the parent strain is not aggregating because of the solvation. That means the solid particles are in reaction with the solvent. Attachment of surface naked bacteria to solid supports. So after these all the tests, they wanted to check if the modified strain is able to attach to any abiotic, that is non-living surface. So they checked for different conditions such as LB, sucrose and glucose. So LB is a rich medium. These are the growth mediums, as well as they checked for different time intervals such as 6 hours, 24 hours and 4 days. So here again, two strains are compared, and we can see that the biofilm formation capabilities as compared to the growth medium are more for the parental strain here also as compared to the modified strain with respect to growth or even for time interval. The parental strain is showing more biofilm forming capabilities. Then the another objective here was to add artificial adhesins. So initially our parental strain has some adhesins. We removed 21 regions from them, and now the objective here is to add synthetic adhesins and check the properties again. So this FOS, and JUN, it represents these are transcription factors, and it is in conjugation with ETAG and IgA beta. So these are present on the outer side of the bacterial surface, and TRX here acts as a control. So then they added this in the plasmids. So these are the plasmid. These are 23880 first and for June also. And after Western blot, it was observed that it was confirmed if they are on the outer surface by checking if adding trypsin is uh, removing these external particles first and June. So in trypsin negative, they are present, and if trypsin is present, they are not there. And conditional flocculation, so they again checked the flocculation of these particles. So they added green and red sensors like GFP and cherry was added for green fluorescence and red fluorescence to both the strains. And then the results were checked. So it was found that 84% of the parental strain, 84% of the modified strain showed they got aggregated, while only 27% of the parental strains were aggregated. Thank you.
Okay. Well, thank you. I mean, you did a very good job, really. Again, your critical appraisal, you think that this is interesting or is just a microphone?
I like this paper, very interesting. Basically, the properties they checked for comparing both the strains, parental and also the modified, those were very interesting. But I found that they could have also checked it with biofilm formation properties also. Like they checked flocculation, that is aggregation before and after. They could have done biofilm formation properties also. So if it is forming a biofilm with some other components like that.
Okay, great. Comments, questions? No. Okay, well, thank you very much. And then we move on to the next speaker. That will be the last of the day. Today? Okay, we'll see how we go the time, okay? So maybe we'll have it tomorrow, but that's all right. We'll see how it goes, right?
Yeah, good afternoon, everyone. I'm Pooja, and today I'll be doing a summary of the paper, a surface display of designer protein scaffolds on genome-reduced strains of pea butida. So, you know that like in the environment, there are many sources of carbon, such as lignocellulosic residues and plastics. So we aimed to like utilize these with the help of an engineered microbe so that we could put them to some efficient use. So for this, we're using pea butida. And why is pseudomonas butida an attractive candidate for this? So we have found that it can utilize various carbohydrates and carbon matter efficiently to produce a useful valoric substrate. And we have also seen that it has other characteristics like its safety status, it has rapid growth, low nutritional demand, various other characteristics. However, it does lack an effective extracellular depolymerizing apparatus, which means that it cannot degrade complex substances alone. So our aim in this paper is to display like a way for it to get an extracellular apparatus for it to degrade this carbon matter. So before I start, I wanted to just explain a bit about the cohesin, Docker, and pairing. So basically, in our bacterial cell, we have a scaffolding subunit on the surface. So the scaffolding subunit has a cohesin binding domain that binds to the Docker binding domain, which is there on the enzymatic subunits. So the greater the binding affinity of the cohesin Docker pair, the greater the effectivity of our enzyme. So moving forward. So for this, we're taking two strains of pea butida, which are EM42 and EM371. So EM42 has 11 non-adjacent genomic deletions, which improves the expression of heterologous genes and enhances its biotechnological potential. But on its external appearance, it basically resembles the wild-type strain, except for the missing flagellum. However, the EM371 has a shaved surface, where all the external membrane structures are eliminated. So the flagella's eliminated structures for developing biofilms, they're all eliminated. So our hypothesis is that the shaved surface proves beneficial for the extracellular recombinant protein production. So let's see if that's actually the case. So our process had 5 steps. So the first step was the preparation of the cohesin containing scaffolding variants and the Docker intact chimeric proteins. So our first gene we took was the SCAF19L gene. However, we found that that was detrimental to the host viability. So we reoptimized the codons and engineered it to get the SCAF19LKT gene. So this gene produced 3 cohesins, the AC cohesin, the CT cohesin, and the BC cohesin. But we saw that the AC cohesin and the CT cohesin had a much higher binding affinity with their respective dockerins compared to the BC cohesin dockerin pairing. So we eliminated the BC cohesin dockerin pairing. So we can see here that we've taken the AC cohesin pairing and the CT cohesin pairing. So for detecting these displayed scaffoldings, we employ the use of 2 tagging reporter proteins. We use the BGIC reporter protein, which is it's an enzyme basically, and it has a very rapid hydrolytic activity. So we found the C terminal of the BGIC enzyme with the respective dockerins, and the same way we did that for GFP as well. So that is our first step. So our next step was a selection of an optimal auto transporter system. So an auto transporter system is basically a monomeric type 5 secretion pathway, which basically is responsible for the display of our recombinant proteins on the cellular surface. However, it is difficult to predict the functionality of a secretion system with a given passenger protein. So we've taken 4 different auto transporter systems to see which one would be best. So these are the IgA protease, the antigen 43 or AG43, the EAA intimin, and ESTP esterase. ESTP was toxic to the host, so we eliminated that, and we checked the other 3 for further testing. So in our test, we cloned the codon optimized gene of the CT cohesin. We cloned the 3 auto transporters, and then this was mixed with the BGIC CT doctrine. So this is what our result was. So we saw that AG43 showed like the maximum absorbance compared to the both EM42 and EM371. Incident, however, it did have an appearance in EM371 but not in EM42, and we attributed that to it not being like the doctrine of it not being able to bind to the EM42 cell surface. However, IgA had around the similar absorbance in both EM42 and EM371. So we concluded that IgA, although the protein was expressed, it was not functionally observed on the cell surface. So now that we've found our secretion pathway, that is AG40, we have to choose the best cohesin dockerin binding out of our AC, CT, and in some cases both AC and CT are expressed. So for this, we used BGIC bound to both AC dockerin and the CT dockerin, and we observed our results. So in graph A and graph B, we can see that EM371 always shows a higher absorbance compared to EM42 in both cases, and we can see that the absorbance for the CT coherent was more than the absorbance for the AC coherent. And another result we saw was that those surfaces which had both AC and CT coherence showed the maximum absorbance. And we observed something similar for GFP as well. So in GFP, A corresponds to the AC coherent and B corresponds to the CT coherent. So again, we saw that EM371 was brighter than EM42 in both cases. And we saw that one with the single cohesin mini scaffolding showed like a less brightness compared to the 2 cohesin mini scaffolding. So our final conclusion was that the CT cohesin doctrine pairing was the best, and even better than that was the cell which had both CT and AC cohesin expression. So now let's try to like quantify the number of doctrine tagged enzyme molecules. So we have like the cohesin doctrine binding as a 1:1 ratio. So if we know like the theoretical number of the BGIC CT doctrine molecules and the number of pre-putida cells, we were able to estimate the number of cohesin doctrine bonds per bacterium. So from these numbers, you can again see that EM371 had more bonds per bacteria compared to EM42, which again demonstrates the superiority of EM371. So now the thing is that, so we have to bind multiple extracellular enzymes to our bacterial surface to degrade like the polymers we require. So although we can display over like tens of thousands of scaffoldings, it's still not enough to anchor all the extracellular enzymes we require. So the thing is, the secretion pathway we have taken, although AG43 is the best, it has not been optimized. So optimizing it will increase number of scaffoldings, as well as removing periplasmic and outer membrane proteases that can cleave the AG43 pathway, or we could even add the secretion promoting serum to the N-terminal display coalescence. And this binding of enzymes also could be further enhanced by adopting a cohesin doctrine pair from extreme thermophiles. And finally, so now we have discussed about like the various factors, the cohesin doctrine pairing and the secretion pathways, but all of these are related to just the binding on the surface. So now we discussed about this display of the recombinant proteins affecting the viability of the host cells. So we've seen in EM42 that the P putida expressing both the cohesins AC and CT had a lesser absorbance than the ones showing a single cohesin. This wasn't present in EM371. So, but however, in EM42, we saw that even though the AC cohesin, CT cohesin, EM42 had a lesser absorbance, it was overshadowed by their greater binding capacity. And another factor we saw was that all EM371 grew slower than EM42. But we are hypothesizing that EM371, although it grows slower due to its greater binding affinity, as seen in the previous cases, will overshadow this. So, and we verified this assumption. So, you can see in the graphs here that when we've taken both the strains, EM371 always grows with a higher optical density compared to EM42. So, basically we conclude that even though EM371 has a lower viability, it still has due to its higher binding affinity, it still is a better option. So yeah, in conclusion, we evaluated the capacities of both strains, EM42 and EM371, and we also saw that all of these cells, they could either display a single cohesin or a two cohesin binding domain. And we saw the two cohesin binding domain, although was expressed lesser than a single cohesin binding domain, did have a higher affinity. So EM371 strain outperformed EM42 in all assays. So this indicates that despite its lower vigor, the EM371 is a promising platform for the attachment of designer catalytic scaffoldings. So EM371, as you remember, was the one with the shaved surface. So this proves that those with the surface shaving demonstrates like a viable strategy for the design of the gram-negative bacterial catalyst. Thank you.
Okay, thank you. Well, thank you. It was a very good summary of everything. So again, don't run again. So your critical view of the thing.
I thought this was like a good first step to like determining how P putida could be used as like an effective bacterial catalyst for carbon sources. But like I think as mentioned in the paper, there are a lot of different ways to optimize this process further. So like I think this is like a great starting point. I think for it to actually be used efficiently, like in a real life setting, there still has to be like a lot more optimization done in like all of the different like factors of it. Yeah.
Okay, great. So questions, comments? Okay, well, so far there haven't been questions from you on the presentations of your colleagues, but so be it. Okay, so I don't know, what do you think? Yeah, okay. I think we should stop at this point, okay? And maybe we take it for tomorrow, okay? Because we still need to have a few minutes to organize for the next papers. Okay, good. Okay, so let me go to the list of those who are, I think that, let me see. I'm a bit confused with so many microphones. Okay, so I think that with this series of papers that I'm going to share now, I think that we complete this series of 4 days where we have basically four presentations of the 4 groups. So what I will do tomorrow after we hear the last presentations is to see who of you have talked and who of you have not talked, and then we'll try to fix that. And well, let's try to wrap up that tomorrow. But for now, let me just say what papers we have here. I don't know whether we can. Okay, well, I have to cross check the names of those that sign up in the different groups versus the names of those who have given me already their presentation. So I don't have that clear, but in principle we have team 1, that was Debomita, Abdul, Ashna, y Tejas. Is any? You are here, okay. So you are team 1, okay. Now Sanchin, Poja, Pranati, Vaishnavi. Okay, you have it there. Team 3, Adita, you're here. And then team 4, Mek, Mutu, and Sunho. Okay, so we have representatives of all group, okay? So please, let's do the following. So paper number 1 goes to team number 1, okay? Paper number 2 goes to team number 2. The 3 goes to number 3, and the 4th goes to number 4. Is that clear? Okay. So the ones that are here, please tell your colleagues that they have to move their ass, basically, and do things properly. Okay, great. And then tomorrow we'll make the final balance of who has talked, who has not talked, and then on the basis of that, we plan for the following steps. So once we see that all of you have the chance to give a real presentation, then we will move on to plan these IEM-like projects, okay? And I really encourage you, if you have the time, to really go into the pages to see how these IEM projects work, and perhaps to get curious about some of them and get an inspiration of what type of propositions you can make. So the propositions that I expect from you have to be completely new. You have to propose something that has not been proposed before, and we will use the last day for, so the last day, that will be on Wednesday next week, I would like to spend that day doing 3 things. One of them, I would like to have a conversation with you about the course, what you have learned, what you have liked, what you have not liked. I would like to have your feedback, not only on the course, but also on the topic as a general thing. And this for me will be very valuable, because as we talk over coffee, I am starting to write a textbook on synthetic biology, okay? And the contents of the book will be very similar to what we have been dealing with these days. And therefore, I would like to know from you what parts you consider more interesting, which parts are more boring, what things you would like to know more, what things you think that you have an excess. I mean, this type of feedback for me will be very valuable. So that would be one thing. The other thing is that I would like you, each of the 4, teams, have to present an IEM-like paper, an IEM-like project, sorry, okay? So that means you have to get together, discuss it a little bit and so on. And I think that you need to know a bit in advance about that the last day we're going to have this IEM-like thing, because you require time to think a little about it. I mean, you cannot improvise one of these projects like overnight. I mean, you have to start a conversation with your colleagues and so, and see whether you can do it. You have to tell me whether you are happy with the teams, or you prefer that now we change teams and perhaps we start a different combination. Okay, we'll discuss that in one moment. And then the third thing that I want to make the last day is to have a little party with you. Okay, so we have a little drink or a little snack or something, and we have to investigate where in the department we can have that, but and say goodbye, and we remain friends, and good luck. Okay, great. So then I think that you know what you have to do for tomorrow. We'll continue with the series of presentations, but then remember that tomorrow will be the last day with presentations, okay? And then after that, we have to start thinking on this other new activity that is about these IEM-like projects, okay? Good. Any question, any issue, whatever? Okay, very good. Yeah, and please remember that I have all the materials of the course in the Dropbox file that I have shared with all of you, okay? So if you need everything is there. So all the presentations of you, all my talks, all the papers, all is there. So you can always return there can find information in case you need it, okay? Good. Any other question? Any other issue? No? Okay, so goodbye and see you tomorrow, okay?